just a normal day, a day to be cherished. Today, we are going to witness the efforts of masterminded students. Yes, it's Agricultural Graduate Students Conference 8 AGS 2023. Theme 3, Emerging Paradigm in Crop Protection. It is headed by Chairperson Dr. Edwin Rockwell Jaising and Co-Chairman Dr. M. Shanti, Directors SCPPS, members Dr. G. Karthikeyan, Professor and Head of Department, Plant Pathology, reputed faculty Dr. Y.S. Johnson Edward Thangaraj, Sir, Professor Entomology, reputed student M. Mr. K. Arvind Raju, first PhD Entomology. Welcoming gains utmost importance in any culture. Now, I call upon Professor, Department of Agricultural Entomology, Dr. Y.S. Johnson Thangaraj Edwards, sir, to welcome the gathering.
Good afternoon to everybody. On behalf of the Agricultural Committee, I'm very happy to welcome
mic on
Director, Center for Conservation Studies. So he is always very keen in explaining all the recipes to our PG students. So whenever we discuss about as a PG coordinator, I know whenever we discuss, we always telling something about the need of the student only. So he is very keen, he shows very keen interest in always providing all the facilities to the PG students. So similarly, also encourage all the PG students to publish their research in high rated or high impact journals. So I welcome our director and I welcome my friend Dr. Edwin Rockwell. So he is actually having more than 25 years of experience. So even though basically he is a lead scientist, he had a rich experience in developing the pesticide molecule. So his lecture will be very much useful to you. I welcome Dr. Edwin. And I welcome our Dean ACRA Kiligalam. So a known pathologist. So he is doing helping very much the PG students in the Kiligalam campus. So in spite of the constraints in the Kiligalam campus, so he is actually doing all his best to improve the facilities there. So I welcome Dr. Theradimani Dean Kiligalam. And I welcome Professor in Department of Pathology. So he is actually helping in developing the pathology department activities. So even our director always that entomology department should develop by seeing the pathology department. So he always says you should come and see the pathology lab. So I think he's developing the pathology lab very much. So I welcome him. I welcome Dr. Bhuneshwari, our professor. So he is actually, I can say, he is the key person in developing the NADL laboratory. So all of you, not only the entomology students from all the departments in the university are benefited by the NADL accredited laboratory. So I welcome him. And I welcome Arvind Raj. So he is our first PhD student. So I know Arvind Raj even before he got here as a SR in Malavachino. So he always talk about research and all that. So he's a student who is very much interested in research. So he always worry about his inability to continue his PhD immediately. At last he has joined the PhD now. Welcome you. I welcome all the students who are going to since today and also all the students coming from other states, and welcome you all once again. And I thank our Vice Chancellor and Dean Speakers for giving me this opportunity. So all of you know, our Vice Chancellor is always showing more interest in the development of our students. So he has actually given you to our TNA students. He has given the scholarship. So for all our MSc students and PhD students are provided scholarship only because of the interest shown by our Vice Chancellor. So similarly our PG Dean also. So whenever as a PG coordinator I approach him, he always immediately ready to help our PG students. So I thank our Vice Chancellor and PG Dean. So once again I welcome you all. Thank you.
That was a soothing word. Before getting words from someone, it is better to know about them, isn't it? It is not just for vanity, but also to inspire the receivers. Now, I call upon Dr. G. Karthikeyan, Professor Head of the Department of Plant Pathology, to give speech about this.
The most respected Director Center for Plant Protection Studies and our beloved Dean, AC&R Curriculum. Today's guest, my friend Dr. Edwin and Dr. Johnson sir, Bhuvneshwari madam, and also Mr. Arvind Raj, my dear student friends and delegates. First of all, I am fortunate to be here for introducing my friend Dr. Edwin. Dr. Edwin Rockwell Jaising is a well-known personality in the pesticide industry, not only in India and also globally. Basically, as Dr. Johnson sir told, a weak scientist, but however, he has a broad knowledge of all the products, all the pesticide products in the world. So currently, Dr. Edwin is working in Cortiva Agri Science, and presently his role is South Asia Applied Science Leader for crop protection discovery as well as development. Previously, he had several responsibilities and roles, which includes South Asia New Commercialization Leader, Global Biology Leader for rice and specialty herbicides, and also Asian Pacific Fungicide and Herbicide Development Lead, and similarly Asian Pacific Data Management Lead, South Asia Herbicide and Fungicide Development Manager, then Territory Field Scientist for Tamil Nadu, Kerala, and also Sri Lanka. Though he has 25 years of service in the field of product development as well as the familiarization, he's a versatile personality, and he has his own competency in the field of crop protection products. Significant experience in product development, data management, technical segmentation, as well as value positioning he has. And he's expert in product characterization and its products. Then he's also expert in project management, about the technical as well as commercial, and he has the capacity to manage geographically diverse team of workers with the team spirit. So with this small introduction, may we now request Edwin to become. So on behalf of the department as well as the university and our CPPS director, we welcome our today's speaker, Dr. Edwin. Welcome. Thank you very much.
Whatever we think about and thank about, we bring about. A famous quote by John F. Dermanti. Now, I call upon Director of CPPS to felicitate the gathering.
On behalf of me, Dr. Bhuvneshwari will continue. Because I need to go for a MOA signing meeting by 3:30 to 4:30.
Now, it's time to honor the dignitaries with a memento. We invite the Director, Center for Plant Protection Studies to present the memento to the lead speaker, Dr. Edwin Rockwell Jaising. We request the Director, CPPS, to present the memento to the Dean, AC&RA Curriculum. We request the Professor in Head, Department of Plant Pathology, to present the memento to the Director, CPPS. We request the Professor and PG Coordinator, Department of Agricultural Entomology, to present the memento to the Professor and Head, Department of Plant Pathology.
Screen off. Background screen off.
We invite Mr. Arvind Raju, Student Organizer Secretary, to present the memento to Dr. Bhuvneshwari, Professor, Agriculture and Entomology.
Now, I request Dean, AC&RA Curriculum to felicitate the gathering.
Please, sir. Open. the outset, I welcome respected Dr. Edwin, chairman of this forum, and members, faculties, and my dear students. Good evening to one another. So, first of all, I thank to BC ma'am for giving this opportunities. On this purpose of this conference, you know that we are conducting so far 7 conferences successfully. Main aim is to share your ideas, knowledge, talent, and also your subject. This is related to pathology. Apart from this, we are having a entomology also. That is a CPPS. So under CPPS, we are sharing that your knowledge with others. So you know that we are India. We are emphasis more on quality of education. Our government is giving importance to quality education. So our institute is such institute which is giving quality education to the students. So when we see the world level, so every country, there is Australia, US and Germany, they are looking for our Indian students. So Indian students are well talented, they are investing more on education towards the Indian education system. They like Indian education system. They know that Indian students are the world the best. They will lead the world. Future of the world is with Indian students. You know that. So this is a such platform. You can utilize this platform and you can share your ideas and everything. Thank you very much.
Now it's time to get some valuable words from Dr. Bhuvaneshwari ma'am, Professor, Agricultural Entomology.
Representatives and professor heads and also those who have joined online and students. So actually I am here to represent Director CPPS. Actually our Director CPPS has to present this felicitation. So because of another objective of attending other program, so I am here to share her views with you about this the guest speaker as well as about this program. So as you all know, that agriculture plays a great role in the economy. As we were student also, we were taught that more than 70% of the Indian population is engaged in agriculture. So it is such an important occupation on par with other industries also. But on the other hand, there are also reports of undernourished people. According to FAO, there are about 795 million undernourished people in the world. So this is a contradictory. That is, agriculture is the major occupation, but in spite of that, we have undernourished or poverty also. So when we see about this agriculture, there are many biotic and abiotic stresses that affect this crop growth and as well as yield. And among this biotic, we know that what is the most important biotic factor? As plant protection students, what we have studied? Insects and diseases as well as nematodes. So all these contribute for crop loss. So the crop loss ranges from 20 to 80%. In some cases, in worst cases, particularly in case of invasive pests, we experience even more amount of crop loss. So what ultimately the farmers are doing to safeguard their crops? So the first line of defense is chemical pesticides. Okay, so which is in line with our guest speakers' profession, is it not? So we always, the farmers, they first prefer to go for spraying. But on the other hand, we are also worried about when we talk about the pesticides, we are also concerned about the environment, is it not? So environment also the health effects. So both need to be balanced. So we should know how to select, how to apply the pesticides in terms of protecting crop as well as protecting the environment. So in this line, the crop protection plays a very key role in protecting the environment as well as and protecting the people and protecting the farmers and food security as well as from food safety point of view, how we apply and how we manage these pesticide application in order to get good yield. And also, recently we are seeing about this invasive pests. So whenever there is an invasive pest, the first tool that we have in our hand is for management is definitely pesticides. So here pesticides play a major role, but we should know how to handle this tool in order to protect the crop as well as to protect the environment. So the current and future pest management tools, if you see, not only the pesticides, but also we are always opting for integrated pest management practices. So that is, we are taught with for many decades about IPM, but still we face problem with some of the tools. Is it not? Except for these pesticides. So one of the, some of the reason tools that we can say about GM crops or RNAi silencing, gene silencing. So there are many tools are coming up, and also from the application point of view also we are now talking more about from pesticide application, UAV, unmanned aerial vehicles. So they are gaining momentum. So now India is formulating rules and regulations. So the framework also we have how to apply these pesticides using these drones. So this is also one of the upcoming technology as well as the research. So to the extent how, to what extent we depend on these pesticides is with the motive of reducing the loss and also with the willingness of the societies to opt for other pesticide management. So in this context, so realizing the importance of pests, the losses they cause, and for the management and application of newer technologies. So this is a right time for organizing this type of session on emerging paradigms in crop protection. And I take this opportunity on behalf of Director, Center for Plant Protection Studies, to convey my best wishes to all the students who are going to present and who are also going to be a participant of this session, and who are also going to display their posters tomorrow to have a very good interaction and be a success in all their endeavors in the years to come. With these words, I have the pleasure to invite Dr. Edwin Rockwell Jaising for delivering his guest lecture on discovery and development of plant protection chemicals. Thank you.
The time we have been waiting for has come. Now we call upon Dr. Edwin Rockwell Jaising, South Asia Field Sciences Leader, Crop Protection Discovery and Development, Cote d'Ivoire Agricultural India Private Limited, to blow us with his knowledge.
Good afternoon all. I thank Director CPPS and Dean here and professors and law students for giving me this opportunity to share some of our process in discovery and development of crop protection products. So almost you heard about me from almost all of my friends here. So I started my career in crop protection industry from DuPont as a sales trainee. Then I moved to R&D. I worked here in Coimbatore, based out of Coimbatore almost for 19 years. And Tamil Nadu, Kerala. Then I moved to the Asia Pacific role, then biology role, and then back to India. I'm leading the India R&D team now. So we have around 11 field scientists distributed across the country, and also we have a field station, research station near Hyderabad. I'm currently based at Hyderabad. So I'll take you through some of the process which few of you may be knowing, or most of us will be learning further today. So first to introduce about Cote d'Ivoire Agri-Science. It's a merger of two different companies, Dow Agro-Sciences and DuPont, and a seed company Pioneer. All three together we have. So Dow and DuPont merged and spin off into three different companies. One is the Cote d'Ivoire Agri-Science. Cote d'Ivoire means heart and nature. So we take care of the nature, and most of our purpose is to enrich the lives of those who consume and those who produce for a generation to come. What do you mean? So whatever we consume and whatever we produce should be sustainable. That means it should be safe, right? Hello. That's our purpose. And next.
Okay.
We have a change it.
Let's change it. Change it. You can use. பென்ட்ரைவ் கனெக்ட் பண்ணிருக்கு சார். பென்ட்ரைவ் கனெக்ட் பண்ணிட்டு இருக்கு சார்.
Okay, no problem. So we are putting farmers at first. Why? We are not studying agriculture, so our first focal point will be farmer. So if the farmer is happy, the society will be happy. That's why we used to take care of the farmers most by providing some of the solution which we are going to bring. As an industry, crop protection industry, we are bringing the crop protection molecules. As Madam explained that the first influence is pesticide, so we are dealing with the pesticides. So, but the reality here, even though our primary focus or report is farmer, so what he is facing a lot of challenges. I don't need to mention everything. So we all know he is going through all these vagaries in his production, right? Next. Whatever we are looking at innovative way to differentiate the solution. Am I audible? Yes, sir. So that the farmer increases his yield, because he will be benefited. If farmer is not able to increase his yield, he will not be able to sustain his agriculture. And if he is not sustaining his agriculture, then he will not be suffering the lack of food. So increasing yield is one of our objectives and enabling farmers to meet consumer needs. We need quality food, right? So days are gone where we can eat anything. Now we are looking for quality food. So we have to, the farmer needs to meet the consumer needs and durable and reliable solution. It's not like today I use and tomorrow I should look for something else. It should be sustainable. And as Madam explained, environmental importance to take care of the environment. So we are looking for to live with less footprint in the environment. Why innovation is important in crop protection? So if you see, crop protection is a chemical, right? Chemical pesticide means side is to kill, CID, right? So we are dealing with the pesticide, so it should have increased selectivity. It should kill only the targeted pest or disease, and integrated pest management. It should integrate with other management solutions, and it should help in the resistance management, right? And novel mode of action. So if we bring the same mode of action because of other factors like cost resistance and all, it may not work for long. So we need to look for a novel mode of action and sustainable food production already I pointed out, and improve productivity, replace some of the older technology which are not safe, which may not be safe, and lower application rate. Why we need a lower application rate? To leave a less footprint or not to leave a load to the environment. So, anyone here able to guess how much money we are spending to bring one molecule? Any idea? Any guess? Maybe in rupees, Indian rupees or dollars? Any idea? No, is it? At least you can say 1 rupee. Why you are silent? Every minute, we need to spend $50 for 11 years. Close to 2,500 crores to bring one product from discovery until the final launch. And the number of years it takes from previously 8.5 years, now it's close to 11 years. I'll explain why it's taking more. And these are all the amounts we have spent, so that we meet all the needs. Like we all heard about the organic, and okay, organic is good, but why to blame others? So we need to prove those with all the safety data. Because of that, we need to spend a lot of money in generating. Like for registration, environmental chemistry, toxicology, and field trials to understand the product performance across all the different environmental conditions, and product chemistry, toxicology, biology and chemistry, all these grouped together. This is 2014 data, around $286 million. Now it's close to $320 million. Multiplied by 80 rupees conversion. So it's close to 2,500 crore rupees. How do we start that one? So, okay, this much money, and how do we know that particular molecule is having insecticidal activity? Before we start that one, we will be looking for some of the natural products, like synthetic pyrethroids. They came from the natural product. we will be looking on some of the natural products and their design and structure. And based on that, we will be looking for some of the synthesis and bioactive hypothesis. Based on some of the chemical, synthetic chemical and hypothesis, we will be looking for some structures and competitive inspired. So a lot of players are here, like you may be knowing, Corteva is one among the top 4 research-based company, like Bayer, Syngenta, FMC, Bayer itself, all those are research-based companies. In addition to that, Indian companies also there. They will be filing patent, we will be studying those patents. So we will be understanding what are the structures patented and across what activity. Similarly, we will be inspired by them also. Then we will generate a lead and we will go for optimizing the lead, and then we will finally release the molecule. So here if you see, as I mentioned here, almost close to 250,000 molecules will be screened initially. So we will get a hit. Then maybe around the 1500 products will have leads, like they may be having an insecticide activity or a fungicidal activity or a herbicidal activity. Then we will prioritize as a project, and 30 project out of 30 project, only one product will be launched. This is how the cycle takes, and it takes from discovery to development stage itself, sometimes it will take 7 to 8 years. Then we have to run fast before we lose the patent. Patent is also important. Okay. Next. So sustainable innovation. Corteva is committed to bring the sustainable product. If you see the sustainability criteria, we have around 12 factors. So first is to zero hunger. In that part, we have to meet the 3 criteria, and on the clean water, therefore 2 criteria. Then overall consumption and production, we have around 4 factors we need to meet. Then greenhouse gas emission and take care of the land. All these 12 parameters, I'm not going through in depth. You can these are all available in the net open. So if you want to know more, you can search the sustainability criteria. All these details you'll get. We will try to bring a product which at least meets 4 of these criteria. So that's what we are committed, and almost the crop protection industry, because of the competition and all, every company needs to meet these criteria, so that whatever we bring will be sustainable. That is safe, and safe to humans, safe to animals, mammals, safe to environment, worker safety, and those who handle and those who finally consume. So these are all the safety parameters we need. And different stages, this is what I mentioned in the stage 0, we will find out that will be multiple products screened. So we will find out whether they have insecticide activity or herbicide activity or fungicide activity. Then we will move to the stage 2, stage 1. Here our concept. This concept means, so we will be looking for insecticide means we will have around 3 to 4 goals. One for lepidopteran activity, then sucking pest and soil-borne and nematicide, motor activity. So like that we will be screening. If they pass on, then we will continue that one. So that's how. Similarly for diseases, we will have homicides, septorial leaf spot, because that's a major important disease in Europe, and is in soybean rust in Brazil, important, and other foliar diseases like alternaria and other leaf spot. So if they work, we will have one. Similarly for weeds also we will have concept like post-emergent grasses, post-emergent broadleaf weeds, selectivity to rice, selectivity to wheat and other crops like that. Then we will move to the development, pre-development stage, then finally development, launch and post-launch. These are the different stages which happen around in 10 to 12 years time. In every stage, we will be looking for those sustainability criteria. So it's a very diverse chemical we are handling, different portfolio like herbicide, insecticide and fungicide. This is the optimization of lead is like right in the middle. So we need a pest spectrum. It should work on the important key pest. Then it should have a potency. It should work at a low rate and it should kill all the stages, weed stages, or nymphs and adults. Like crop safety is very, very important. Mode of action, it should be new, as I mentioned earlier. Mobility in the plant, whether it's mobile or stable, because based on that, we can target different pests and business case. It is for the company to decide whether we can advance that molecule or not. Then ecotox, field translation, some of the time, initially it will show activity in the lab condition. When we move to the field, it may not have that activity. So then finally formulation and metabolism and soil persistence. With all these, when it passes through this, then finally we'll start working on that one. So until now I talked about how we screen. Then now I talk about some of the regulatory challenges, registration challenges, which take care of our safety and environmental safety as well. So every regulatory environment is complex. Like European regulatory environment will be different when compared with the US, and India will have our own requirement. So we need to generate all those studies to meet other complexities. See, for example, any molecule, whether it's a chemical or a biological or natural product, they will have a metabolite. We need to generate the data on those metabolite also. Otherwise, we will be focusing only on the active ingredient, and those metabolites may not be safe. Similarly, for every country also, they will have different regulatory challenges. This much, see, this is the while we are sitting here, which the data package we are submitting for a product to register. So this much data we need to generate. As I mentioned earlier, it will need to have the product chemistry, biology, toxicology, and environmental safety, everything. So overall, 150 studies like product characterization and human risk characterization, then environmental risk characterization, then regulatory submission of codes. All these studies are needed, and as I mentioned, country to country, the requirement will be needed unique. So we need to generate country-specific data as well. But how we are claiming that we are bringing a safe molecule or a safer molecule? So we have a reduced risk criteria. Here we are looking for a product which are having, especially mammalian toxicity. We are all mammals, right? So there should not be any genotoxicity, no immunotoxicity, then neurotoxicity, subchronic toxicity, chronic toxicity, carcinogenicity, reproductive toxicity, and low acute toxicity. These are all the things the new molecule, the new chemistry should meet before it passes on to that one. So that's what we call reduced risk to human health, reduced risk to non-target. It should be beneficial to the, it should be safe to the beneficiaries, or it should not target the neighboring crop or neighboring other animals or living organisms. And groundwater contamination, it should not be mobile, it should not go to leaching, and it should not go to groundwater contamination, and it should be compatible with the integrated pest management. These are what we call reduced risk criteria. And we have green chemistry classification also. I don't know how many of you gone through this green chemistry. We have a kind of full criteria, so that any chemistry, not only for crop protection chemicals, any chemical synthesized, if they pass through this, they can call them as a green chemistry. So we, for agrochemical also, we have this green chemistry classification. So which helps us to bring a safer or reduced risk products. Okay, next. So here, whatever how we deal with this, our sustainable approach is one on land and environment. Here, favorable tox profile and environmental toxicity. Then on communities, communities means beyond the society, like pre-harvest interval. So how long should be the gap before we last spray and final harvest? Longer the period, it's safe or unsafe? What is needed? You need a longer PHA or shorter PHA? So we will be looking for a product which has shorter PHA, especially vegetables. We have multiple pickings, periodical picking. For that, PHA is important, and broad spectrum at low use rate. Higher use rate means, as I mentioned, it will load the environment, and farmers, they are handling, right? So it should be safe, so that minimum PPE. PPE is personal protection equipment, like wearing mask, gloves, goggles. I don't know how many of the sprayman using today, but this is important IPM program, short interval. That is, I spray today, whether I can enter the field same day or not. That is a shorter interval. For some of the older molecules, conventional molecules, if we go by the label, they will have minimum 2 days of entry restriction also. Right? So nowadays, after 2 hours also we can enter. So then IRM strategy, risk management strategy also. But these are all the approaches we are taking to sustain this one. So how this reduced risk chemistry classification, or how, if we follow those criteria, how it is helping us? So one example here, in US, the normal one, this amount closer to $800,000 we need, and 36 months to process that one. But with the reduced risk category, we are minimizing it by 2/3. So 24 months they will be able to process. And now there is a recent development. If the product, new chemistry meets all these classifications, whenever they meet the same day, they can take a decision also in US. So that's the kind of provision which helps us if we target a reduced risk chemistry plus the green chemistry classification. Next. On another example, Brazil. Brazil normally to develop a product, after generating data, after submitting, it will take minimum 7 to 10 years to scrutiny. By the time most of the countries will lose the patent. But we have an example, one of the herbicide Rinska, I'll be talking a little bit about that one. And Zorvak, it's a fungicide. We submitted with this all safety data and all, we cleared it in the faster time frame. Within 3 years, we got the clearance. So these are all the advantages of reduced risk, or this is how we are managing so that our environment is protected. Again, the same, almost it's a kind of repetition, how the farmer is benefiting, especially tolerance exemption. Tolerance exemption means whenever we export a product, so it should meet the importing country's MRL requirement also. So it may be registered in India. If the product is not registered in Europe, if we are exporting grapes, it will return back. If we export basmati, it will return back. So by bringing a reduced risk or green chemistry product, farmer will be benefiting by this one. You don't need to wait for the other country to register the product. And again, both are same, eliminate the maximum permissible levels and reduce concern of residue of the crop, and minimum PP already mentioned, short area entry and IPM and all. So government agencies, I explained with the example US and Brazil example, and environmental also, it's completely safe because of the reduced risk. So when coming to Koteva, we have got 6 times the Green Chemistry Award. This Green Chemistry Award given by American Chemical Society in US. We got 6 times this award, an industry award, a crop science award, National Association Manufacturer and R&D 100 award, and also recently the reduced risk designation. So we got 20 plus years of experience put together from both the legacies, and we got this award for 6 times. So what we are trying to bring here, whatever we bring, by nature we will be looking for the safer product. I will give some examples. So this is a recently launched rice herbicide. This is a very low use rate, 30 gram active protected, and formulation with high proportion of green solvents. We developed the formulation UC formulation. Entomology students may be knowing that one UC is emulsified concentration, you need a solvent, mostly it's a petroleum primer. So here we developed a product with the plant-based solvents. Globally they will be called Neo-EC, N-E-O, Neo-EC, but in India there is no classification, so still we registered EC. And best in class regulatory profile. It is very safe. How it is safe? Even in aquatic ponds also it can be used, safe to fish, and it will be safe for us. It's a potable water also is registered. Then exempted from tolerance and favorable environment and designated as a reduced risk. If you see, agronomy students will know this one. Propanil, the earlier use rate was 3 to 7 liters per hectare. Then 240 around 500 to 1800 gram active. Then triclopyr 160 to 240. And Rinskar is 30 gram active, 10 to 30 gram active. 10 gram active is enough for aquatic weed control, and 30 is for rice. So this is the way we are reducing the use rates, and also we are looking for a safer product. The another example is Genuelva active. So normally we call the basic discovery company, they will give a trade name for the active also. Trade name is normally the final formulation. So we give a trade name for the active also. Rinskar is the active name for 4-benzoxy benzene. Then Genuelva is pinoteron. So in the market you have delegate, in the trade name of the final formulation is delegate, and the active ingredient is pinoteron. Genuelva means active ingredient. So after the patent expiry, generic product will come. no one can use Gemelva, we can still use Gemelva. Spinotram will be used, they can use Spinotram, but they cannot use Gemelva. So that to differentiate between the basic owners versus the generic, we are giving naming with the active also. This we all know, we don't need to, this is naturally inspired product, and green chemistry award, R&D award is one of the why we have given Palamilum example. Palamilum is a devastating pest, invasive pest, as Madam pointed out. It was introduced in India during 1718. We observed this one. It was in a rampant stage in 1920. Delegate at very low use rate, 100 ml per acre, was able to control this one. Next. For example, it's a reclaimable active. It's a nematicide. The trade name will be Salibro. We just got the registration. We will be launching this product in this year. It is pure nematicide, contact action. It's not having any other activity. So it's compatible with the other living organisms like biological. If you are applying with any of the soil biological organisms, it is compatible. So it's takes care of the soil. This is soil health. So it's this is also one of the sustainability criteria I mentioned. Whatever we bring, it should protect the soil. Next. And natural products, as I mentioned earlier, Kotava is a long 20+ years experience in green chemistry, as well as natural product discovery. We are the leaders. So I already mentioned about the Spinotram, which is Gemelva active, and we have Spinosad. The earlier trade name, the current trade name was Tracer final formulation, Tracer, and Spinosad is the active, and we call them as Qualcova. Then also we have two more fungicides, Inatrac and Ravel. Both of them are working against Septoria. It's registered in Europe. Inatrac already launched, and Ravel will be launched this year in Europe. As I mentioned, the cost is also important. So it's a very low use rate, around 50 gram active. It is very good for Septoria, but for other diseases, it may need 3x or 4x time. So we will not be able to use this until we find a process to minimize the cost. So it may not be available for India and APAC countries, but it's very effective against Septoria disease. Finally, the biologicals. So you may be wondering, because I saw the 10 presentation list, all are natural or biologicals, right? So we also work on biologicals. So as I mentioned earlier, we worked on naturally inspired products. 4 products we developed and two more in the pipeline. Now we acquired 2 companies. One is Solar, another one is Simba. So by this we got access to the biologicals also. If you see biologicals, we can group them into 7 categories under 2 different headings like biostimulants and bionutrition plus biocontrol. We have nutrient management, biostimulants, and soil health and water use. They may be either enzyme based or amino acid based, or some of them are microorganism based. And under biocontrol, we have biofungicide, insecticides, pheromones, and we forget this also. Biologicals overall we can group into 7. All the 7 we have globally, and India, few of the regulatory challenges are there, especially for the biocontrol on the bringing for insecticide, nematicide, or biofungicide. If we bring a microorganism which is not present in India, we may be having a challenges to bring that product. But we are committed to bring those biologicals also. By this, we will create a valuable biological business. So this will go in combination with the chemical solution. Wherever there is a high pest pressure, and we all know biologicals will have a limitation, where we can go with the chemical way of controlling, and wherever we can manage from the beginning systematically, we can manage with the biologicals. So we have all the solutions. So by this, I think I covered how the chemicals are started from discovery to development, and how we are meeting all the regulatory challenges, how we are able to manage the sustainability, like safe to animals, mammals, humans, and also the environment, and overall how we are balancing between biologicals and chemicals. So this, since I am from Koteva, I gave some of the examples from Koteva. If you go and look into any industry, like basic research based companies, they will have this kind of wide spectrum. Like if you take about Syngenta or Bayer or FMC and BASF, everyone is committed to this kind of safety and everyone is committed to bring biologicals also. So it's not a thing of the past or only the privilege will get the biologicals. So we are having the biologicals also to meet all the common. So I think by this overall it's already summarized. So any questions? I am ready because some of them I covered very top level because it may not be needed for all. So if anyone wants to ask any specific questions, I am ready to answer.
What is the procedure for bioproduct?
Yeah, that's what I mentioned. Some of the for biostimulants or enzyme based or amino acid based, we have the guidance, regulatory guidance. For biologicals, like if I am bringing any microorganism which is having a fungicide activity or an insecticide activity, if it's a native strain, still we can do that one. We have to show similar to our crop protection chemicals only, 2-season bioefficacy plus residue and the soil persistence and all, and then formulation and all we got to disclose and we can do that. But for a company like us, multinational, if we have a strain or a new species found outside India, and it's having better activity or efficacy compared to the Indian strains or Indian species, there is no guidance. So as I mentioned, we purchased 2 companies. We have 2 promising, one is biofungicide and one is bioinsecticide. But the challenge is one is the new species in India and one is new strain in India. It's not yet reported. So we don't have any guidelines as of now. So we are working with the government agencies. Otherwise, for the biostimulants, enzyme-based or amino acid-based, we have the guidelines. Similar to the crop protection.
Duration also same.
Same, right. So in India, we need to generate 2-season data, so 2 years gone. Then after submission, they take 2 to 3 years. So minimum 5 years to bring a product after we start the. Okay. So normally they say there's no question means that way understood or other way. I don't know. It's after lunch, maybe session. Thank you for the opportunity and as I asked about this kind of what you call conference or seminar, whatever. So I heard for the first time, or I inquired more about this thing. So you are all organizing this very good conference. So make use of it. So learn from each other, and if you are yourself inviting people like me with the support of faculty members and dean and directors, so make use of to learn more, and we have a lot of opportunity. And on a company side also, that's a big opportunity. It's not necessarily you should look only on the state university because you are all PG and PhD, right? So don't think that there is no opportunity outside. We have also opportunity. And recently I hired, not for Tamil Nadu, but for other geography, around the last 3 years I hired 6 field scientists across the country. So lot of other companies also having the opportunity. So develop your talents, and only one thing is you are all doing research. Think what is good for us, for the farming community and for the society, and work on that topic, and don't focus just to publish. What next? How our finding or my finding will help the farming community or will help the society. So think from that angle also, start working on that one. Though for MSc or PhD, it is just a research or a kind of credit requirement, don't look into that one. How we can take that research. Based on your support, your guide and your faculty members will be helping you to do more. So it's all with you to bring more product. Right? Thank you. Thank you.
Thank you, sir. Now, I call upon Dean Kirikulam to give memento to first PG coordinator entomology. Announcements was excellent. Now, I call upon Mr. K. Aravind Raju, first PhD student, to propose a vote of thanks.
Very warm greetings to everyone. Attitude of gratitude is a inevitable good quality that one possesses. I am very glad in presenting vote of thanks for the three sessions, emerging paradigms in crop protection. I am Mr. K. I take this opportunity to thank Dean SPGS for organizing this Agricultural Graduate Student Conference 2023 in a grand and successful manner. I thank Director Center for Plant Production Studies who graced this occasion with her presence. I render my sincere thanks to our team hero, our chief guest, Dr. Edwin Doorbell Jaising, South Asian Field Science Leader, Cortiva Agricultural Science India Private Limited for his presence and wonderful lecture. I thank Dean Asian Agriculture College and Research Institute Kilikulam for his presence and felicitation during this session. I sincerely thank Dr. Bhuvaneshwari Madam, Professor Agriculture and Entomology for their presence and felicitation. I thank Dr. Johnson Edward Kandaraj, PG Coordinator, come AGSC Team 3 staff representative for his guidance for this session. Not but not least, I thank my Team 3 session members Vishnu, Priyadarshini, Monisha, Mohana, Praveen Joshi, Dharini, who made all the arrangements for the session in their own. Also, I personally thank all the persons who are all behind this successful session. Thank you.
Thank you, Mr. Arvind Raju. Now, let's move on to the most important part of the session, oral presentation. We invite the first participant, Ms. B. Kirtana, to present the topic, Influence of Weather Parameters on Seasonal Occurrence of Broad Mite Polyphagotarsonemus lattice on chili.
A gentle reminder to the presenters. Presentation needs time management. It should be clear and crisp. 7 minutes is allotted for presentation and 3 minutes for discussion. Your presentation will be valued based on the content of presentation, clarity and flow, and ability to interact with the students and the management of your precious time.
First, coming to introduction, first chili is an important vegetable crop and if that alkaloid which is a grasses which attracts many pests and it has an important medical properties. And coming to that, in chili there is two important non-insects, that is the chili mite and thrips. Here I am going to deal with chili mite, that is it is also called as broad mite, chili mite, yellow mite or Moroni mite. The symptoms which includes the downward curling, a leaf with elongated petiole where the buds and stalks get distracted and the rusty appearance in the fruits was observed. Here I have observed the dynamics of the list 18 and hybrid cove chili that is in auction. Here the five plants which were selected randomly in the field from where the observation was taken from top, middle and bottom of the leaf. From the top, middle and bottom of leaves, 1 square centimeter area has been selected in the leaf and the observation was taken and the test count was noted down. This is the observation which has been taken from 46th standard week to 24th standard week, standard meteorological week, that is from November to mid-June. It was observed that during the 46th standard meteorological week, that is during November, the low mite population, that is about 0.24 numbers per centimeter square has been observed. The weather parameter which was correlated with the weather parameters which includes maximum minimum temperature, morning and evening relative humidity, rainfall, wind speed and sunshine mass. Whereas the highest, the peak incidence of mite population was observed during I think it is around the March, that is 3.89 numbers per centimeter square of mite was observed. Here the 10 plants was randomly selected and the observation was taken and the mean average of each standard peak was calculated. The correlation analysis between the weather parameters was calculated. Here the weather data was collected from Agro-Meteorological Department, that is TNAU. Here it was seen that the maximum temperature and sunshine was significant and it is positively correlated with the chili mite population. Whereas it is negatively significant and negatively correlated with the morning and evening relative humidity as well as in rainfall. By using regression analysis, it was observed that the R-square value is about 0.723, that is 72.3% of the pest has been influenced by the weather parameters.
Questions? The session is open for discussion.
Okay, you mentioned that
November population and March population.
Correct, sir.
How you will convey this to farmer and how he will make use of this data?
During summer, the population will be higher in mine, sir. So that water can be sprayed, because if water sprayed, the temperature gets the temperature gets lower, so the mite population can be controlled.
Okay, so can you think about the practical feasibility of spraying water? So how he will be, or how to convince him to make use of this?
Sir, maybe such as in greenhouse means we can use sprinklers, sir.
Okay. Yeah, thank you.
Gentle reminder to presenters. Presenters, please do wait after you present. We need to capture the.
Upon the next presenter, Mr. M.S. Haran, to present the topic, an insight into odorant binding protein 4, precursor of Apis mellifera.
Insight into odorant binding protein 4, precursor of Apis mellifera. Here, about the odorant binding proteins. Let me discuss about the odorant binding proteins. It is a protein which primarily receives the volatile cues, which and aid in the chemosensing mechanism in insects. It is primarily expressed the gene of odorant binding protein 4 of Apis mellifera. Shown its higher expression among other genes expressed during the hygienic behavior activity of honeybees. Hygienic behavior activity is an important form of social immunity in honeybees, in which the diseased parasites and abnormal roots. In the case of parasites, means varroa mite is a major problem in honeybees, which is a mite pest of honeybees causing colony loss. During this hygiene behavior activity, the groups being sensed abnormal will be removed by the nurse bees. This chemoreception will be based on the physiochemical properties of this odorant binding protein and the volatile cues emitted by the parasitized brood. With this background view, the objectives of the study focuses on structural and functional characterization of this odorant binding protein 4 of Apis mellifera, and also how this odorant binding protein 4 of Apis mellifera interact with the hygiene behavior related volatiles emitted by the diseased and parasitized brood larvae. The materials in this protein analysis, there is two parts are involved. One is structural and functional analysis. In the structural analysis, the studies involve the primary, secondary, tertiary, and tertiary structure analysis and their topological prediction. Whether the protein is extracellular and the intracellular transmembrane protein, etc. Like the studies are involved. In the case of functional analysis, the protein-protein interaction, whether other proteins are involved in the hygiene behavior activity when this odorant binding OBP4 is expressed. Then in the case of docking studies, or in the case of docking studies, the molecular interaction mechanisms is studied between this hygiene behavior volatiles and this odorant binding proteins. Primary structure analysis. Primary structure is the individual amino acid residue present in a protein sequence. Concerning this OBP4, it is glutamic acid rich protein. This glutamic acid contributing for the 11% of this OBP4 protein, and there are about 136 amino acid residues present in this OBP4. Then another important parameter is the theoretical isoelectric point. It is a pH at which the electrostatic potential of this protein is 0, that is net charge is 0. Then another important aspect is instability index, where it should be less than 40. In the normal environmental condition, it is a stable protein, which depicts that it is a stable protein. Then GRAVI, that is grand average of hydropathicity. If the value is negative, it is more of a hydrophilic protein. If it is a positive, then it is a hydrophobic protein. The result shows that it is a hydrophilic protein present in extracellular fluid. Then secondary structure analysis. Secondary structure means of a protein means alpha helix, beta sheets, and random coil. In this protein, it is predicted to be the predicted that it is majorly composed of alpha helices, and this topology prediction by Proter it shows that the first 20 residues contribute for the signal peptides, and the whole protein is present in the extracellular region, which is a non-bound protein. This TM-HMM probability and target 2 predictions show that there are in TM-HMM, it supposed the prediction that it is a non-transmembrane protein that is present in extracellular fluid. Then the target P predicts that it has signal peptide in the first 20 amino acids. Then there is structure analysis. In this study, we have developed a model based on homology modeling method. Different types of representation of the proteins are presented here. In this electrostatic surface represent that it is a negatively charged protein, which means that red color is negative charge and blue color is positively charged. More of a red color shows it is a negatively charged protein. Then the hydrophobic surface, yellow color is hydrophobic nature. Since it is more of a hydrophilic nature, with some hydrophobic attributes are also present, where that hydrogen, there is a hygienic behavior volatiles are supposed to bind in the hydrophobic surface. Then protein-protein interaction. These are all the other proteins expressed, co-expressed during the expression of OBP4 gene, and these are they all involved in the olfactory reception process. And as I already said, based on these gene ontology studies, it is an extracellular component, and the main protein domain present in this protein is pheromone or general odor and binding proteins. The molecular interaction mechanism. Several hygienic behavior volatiles have been focused on the study, which are specific for varroa parasitization. Among that volatile cues, 2-nonacasonone, that is a ketone molecule, which shows higher binding energy and more the lowest binding energy with higher binding affinity. It interacts with the tyrosine 80 of 3onine 80 of this amino acid protein. 3onine 80 amino acid of this entire protein. Then research findings: OBP4 is a stable protein with 15 kilodaltons of molecular weight, and it contains a signal peptide, and it's it has no transmembrane helices. It is exists in extracellular fluid. Then this varroa specific among the varroa specific volatile cues, 2 primarily induce this hygienic behavior. Thank you.
So who will use this data?
Sir,
or for whom this will be useful?
Sir.
Okay, my tone was different. So for whom this finding will be helpful, how we can take it next time?
Sir, as like you explained earlier, it is an earlier phase of like that of pesticide molecule screening, in vitro screening. These volatiles can be synthesized analogously, so that we can generate chemicals. We can synthesize chemicals that specifically induce hygienic behavior when we apply topically in the honeycombs, especially since they are particular to the specific pest. The target specificity is also here. It is self-explanatory, sir.
As asked by the chairman, I also have the same doubt. This is a very basic study. And what is its application? That's what we also wanted to know. What is its application?
Hygienic behavior activity is one specific trait posed by honeybees, which is a disease and pest resistant factor. These volatile cues play a major role in inducing this hygienic behavior activity. So if we induce like biostimulants, if you apply these pheromones or volatile cues as a product, we can induce this hygienic behavior activity in the case of pest infested or disease infected honeybee colonies, thereby combating these pest and diseases.
So it's an alternate method for management.
Yes, ma'am. Yes.
Hello. So how do you differentiate this protein with the other protein?
Sir, based on the differential gene expression of hygienic behavior activity the honeybees performing hygienic behavior activity and which are not involved in hygienic behavior activity. Several genes have been reported, sir. They hike in expression during this hygienic behavior activity. Among that, these showed that this odor binding protein 4 topped the results. In about four reports show that odor binding protein 4 is majorly involved in this hygienic behavior activities. With that, what about the R protein? Sir, R protein, they are not majorly discussed. Other proteins are also involved, like cacophony proteins, which are R-like proteins, in which their leucine-rich repeats are present.
Thank you, Mr. Haran, for your mind-blowing presentation. We invite the next presenter, Mr. K. Shivanyanaparam, to present the topic, Characterization and Documentation of Begoma virus.
Good afternoon to all. I am going to present my topic entitled the Characterization and Documentation of Begoma viruses infecting cucurbitaceous crops in Tamil Nadu. That's coming to the introduction. Bitter gourd, ridge gourd, ivy gourd, pumpkin, cucumber and watermelon. These are the major cucurbitaceous crops cultivated in Tamil Nadu, which belongs to the family Cucurbitaceae. In India, gourds and melons are cultivated in an area of around 1.4 lakh hectares with a production of 2,491 metric tons. In Tamil Nadu, the major cucurbits growing districts are Coimbatore, Tirupur, Erode, and is followed by Dindigul districts. These cucurbitaceous crops are affected mainly by DNA as well as RNA plant viruses. And the RNA viruses, which includes cucumber mosaic virus, potato ring spot virus, potato leaf roll virus, and cucumber mild mottle virus. In case of DNA viruses, the major viruses which infect cucurbitaceous crops include tomato leaf curl nudity virus, squash leaf curl China virus, bitter gourd yellow vine virus, which was followed by coccidia mosaic virus. These DNA viruses which affect the fruit set as well as the quality as well as the production of the cucurbitaceous vegetables, which also causes the yield loss up to 100%. Coming to the introduction about the Begoma viruses, Begoma virus is nothing but the bean golden mosaic virus, which comes under the family Geminividae, which is derived from the word Gemini, which contains the twin particles, which contains the genome of both DNA A and DNA B in their genome. The shape of the Begoma virus is icosahedral in shape, which contains 110 core proteins, subunits organized in 22 pentameric capsomeres. It is a circular single-stranded DNA genome. The size of the virus is around 2.5 to 2.9 kb in size, and the particle size is around 22 into 38 nanometer. These Begoma viruses contains the 9 genera. Among the 9 genera, the 68.1% of the Gemini viruses belongs to the Begoma viruses. Till now, there are 322 virus species are documented under the Begoma viruses. These viruses are mainly transmitted by whitefly, especially the Bemisia tabaci. The rowing survey was conducted during these 3 major districts, Coimbatore, and Dindigul districts. Among them, the Coimbatore in the Pollachi areas, the Tapatakalam and Puttur, which recorded the highest disease incidence of up to 100% in bitter gourd crop. And the lowest incidence is recorded in bottle gourd, which is recorded in 8%. The symptoms of Begoma viruses infecting different cucurbitaceous crops include: in the ivy gourd it shows the yellow mosaic, in bitter gourd it shows leaf curling symptom, in the pumpkin it is a yellow mosaic, in bottle gourd it's a leaf narrowing symptom, in cucumber it's yellow mosaic, in snake gourd it's a mosaic mottling, ridge gourd it's a yellow mosaic, in watermelon it's a puckering symptom. Then the DNA was extracted by the CTEM method. The infected leaves which was ground by using CTEM buffer, then subjected for centrifugation, which was followed by PCR, then sent for sequencing. Then the Begoma viruses are amplified using this specific primers, that is the Rogers primers, which is universal. The randomly 10 selected samples were subjected for PCR, and the research shows that it has amplicon size of around 1250 base per, and the LAN 12, which shows the positive control, which is a chili leaf curls symptom. 10 positive samples were sent for sequencing, and the accession numbers were obtained for all the 10 study isolates. The majority of all the 10 isolates match with the tomato leaf curl nudity virus. Based upon the sequencing results, the phylogen tree was constructed using neighbor joining method. Among all the 10 isolates are matched with the tomato leaf curl New Delhi virus, which also share which match with the already submitted tomato leaf curl New Delhi virus in the NCBA database Genbank sequence. From this, I conclude that the entry of tomato leaf curl New Delhi virus is of recent occurrence in the cucurbitaceous crops ecosystem. Among the viruses, the begomoviruses, tomato leaf curl New Delhi virus infects around 97% of the cucurbitaceous vegetables, which was followed by bitter gourd yellow vine virus and then squash leaf curl virus in Tamil Nadu. Because the reason behind the tomato leaf curl New Delhi virus occurrence is due to the recombination, mutation and reassortment in the virus genome. Compared to the plant RNA viruses, the DNA viruses, especially in the cucurbitaceous crops, is threat for the cucurbitaceous cultivation. Apart from this virus, the white fly, particularly the biotype B biotype associated with this transmission, due to their high fecundity behavior and the host adaptability, the virus is highly transmissible in the cucurbitaceous crops. From this, I conclude that the accurate and early diagnosis is important for the successful disease management in the cucurbitaceous crops ecosystem.
The session is open for discussion.
Included with early detection. So, how will you pass on this to the farmer?
Sir, just we have to characterize in one, for example, in particularly in bitter crop, there if you have one virus means it will be easy to diagnose. There are combination or mixed infections of 2 or more viruses. That is, it's very difficult to even for characterization in the virus parts. That's the difficulty.
The other part you conclude is management. How will you manage? Or farmer?
Sir, for growing any resistant varieties.
Because there is no presentation. Who is the vector here?
So we can manage the vector, right?
Yeah.
It's a novelty of your research work.
Sir, we have selected majorly focus on 5 or more different cucurbitaceous cards, like bitter gourd, snake gourd, pumpkin. Yeah, the blend, sir. No, sir, for characterization. Other than
everything is over.
Okay. Then apart from TOLCD, is there any other viruses combined with that?
Any virus is there? New virus is there?
The bitter gourd yellow vine virus and the squash leaf curl virus.
So what is the status of
Sir, brinjal I didn't only cucurbitaceous. Yes. Sir, both is there, sir, mixed infection.
Thank you, Mr. Shivananapalum, for your wonderful presentation. Next, we call upon the next presenter, Ms. Kasturi Sharma, to present on the topic efficacy of certain newer insecticides against termite, Odontotermus obesus, and red ant, Dorylus orientalis.
Good afternoon to everyone present here. My name is Kasturi Sharma. I am from Assam. So this was a part of my master's research. So the topic is on bioefficacy of green chemistry insecticides against termite, Odontotermus obesus, and red ant, Dorylus orientalis. So termites as well as rodents are a serious pest in Assam. So these termites are a major pest. So these termites are a major pest in the tea gardens of Assam, and the infestation that is caused by termites is estimated to be around 90% in the tea gardens. And also they cause a very serious infestation in the sugarcane industry also. And likewise, radon is also a serious pest of the solanaceous and cruciferous crops. And these radons used to cause damage to the tubers of potato, in which small minute holes are produced, and which reduces the quality of the potatoes. So because of its subterranean nature, it is very difficult to manage the space. So the objective of my research was to evaluate the efficacy of certain newer insecticides against termite, odontotherapy services and rated Droelis orientalis in laboratory conditions. So the termites were collected directly from the live mounds using Kurpi, and then they were screened in the under the microscope. So we have used a worker cast, since the worker cast is mainly responsible for causing damage, and so the radiants were collected using bait traps. So in the bait traps we have used banana as well as sugar crystals to attract the red ants, and they were installed in the potato crop, and in earlier we have used jaggery and ghee, but the thing is that the radiants used to get drawn in it and die. That's why we have then used these sugar crystals and banana trap for attracting these radiants. After this, these radiants were cultured in the laboratory using a cylinder where we have placed sterilized soil, and then we have given potato as a food for the radiants, and the radiants used to be alive for about a month. So here are the details of the biocide test. So 3 insecticides, imidacloprid, clothianidin and thiamethoxam were evaluated using this dry film method. So in this dry film method, what we did, we took 1 ml of the insecticidal solution in the bottom inner surface of the battery plate, and then we have soiled it. After that, when the toxicant film is formed, then we have placed 20 termites per petri plate and 10 radiants per petri plate. So these were the treatments. So in case of termites, as you can see, the concentrations were 1 ppm, 5 ppm, 10 ppm, 50 ppm and 100 ppm. And in case of radiants, the concentrations were 10, 50, 100, 500 and 1000 ppm. So we have used this distilled water as control. So these are the data at 50 values. So we have recorded the mortality rates after 6, 12, 18 and 24 hours, and then we have used those mortality data in SPSS software to find out the LC50 and LC50 values. So here we can see that thiamethoxam was the most effective insecticide out of the 3 insecticides that were tested, and in case of L50 value also we can see that thiamethoxam was the most fast-acting insecticide. So in case of radiant Droelis orientalis also we have found similar results. So thiamethoxam was the most effective insecticide, and also it was the fast acting insecticide compared to the other 2 insecticides. So this is the conclusion. Among the 3 insecticides that were tested, thiamethoxam was found to be the most effective one and the fast-acting insecticide compared to the other 2 neonicotinoid insecticides. Thank you.
Now the session is open for discussion.
So these are all lab studies, correct? So if you want to recommend to the field condition, how will you bring that? Any correction? Sorry.
So we have to study in the field conditions so that we can know the current rules that can be applied in the field to control the space.
So how you will propose that study? You have thought about that, or now you did the lab study. So if you want to do a field like for termite, you start with 5, 10, 1, 10.
10, 20, 20.
Right. So and multiple 10 factors for a time. So similar to that, if you want to work out some dose at the field, how will you start? Did you thought about that? Because already Actava or imidacloprid or other product is available in the market. This can be really recommended to the farmer as well based on the current. Did you thought about that one? How to work out the field? dose?
We need to do further studies so that we can know about their efficacy as well as their influence on the non-target organisms or beneficial insects. We need to study those properties of these insecticides also so that it will be safe to the environment.
So what are the non-target organisms you will try?
So, beneficial insects like honeybees.
But these are all soil.
Soil pesticides, but it can be also applied as foliar as well as acid treatment also. And they are systemic insecticides. That means it will be absorbed through the roots and it will be delivered to all the parts of the plants. So it may indirectly affect the honeybees also.
Thank you, but again, the major crop you mentioned is students.
Yes, sir. In case of Assam, in my place, these termites as well as these rodents are very serious pest to the tea gardens. But nowadays, in case of tea gardens, one more pest has evolved. It is the green semilooper. So it has developed resistance to most of the insecticides. As one of my friends working in the tea garden, so he told that this specific green semilooper, it has developed resistance to most of the insecticides and it is very difficult to manage.
What is the purpose of your study?
Sir, mainly to find the LC50 and LD50 values of these insecticides and find which was the most fast acting and the most effective insecticides. Yes, sir.
What about field? Field by
The field between you, our research was just limited to laboratory studies.
what is the insecticide recommended by farmers?
Mainly in case of termites, nowadays what we are following is fipronil and chlorinated insecticides.
What about chlorophyll? Chlorophyll.
Chloropyrifos. Earlier they have used this chloropyrifos, but currently they are using this fipronil and chlorinated.
Thank you, Ms. Kasturi, for your wonderful presentation. Refreshment is provided outside the hall. I request everyone to take a break and assemble back sharply after 10 minutes. Attendance for this theme will be issued after the break. Let's move on to the next presentation by Mr. M. Ayandurai on the topic Fusarium dieback and shortwood borer, symbiotic infection, a major threat to avocado production in Tamil Nadu.
Another present here. I am going to present my oral presentation about Fusarium dieback and shortwood borer, symbiotic infection, a major threat to avocado production in Tamil Nadu. So in this particular presentation, I will clearly explain why the Fusarium dieback is a major threat now avocado cultivation in Tamil Nadu. So and also what is the mechanism behind the symbiotic infection of Fusarium and the insects. First of all, avocado is a highly nutritious food. It has a good fat content. It means they are having a 30%. In compared to protein, it's a 4% only protein content. It cultivated in our Tamil Nadu, Kodaikanal, Erkadu, Coimbatore, and Ravapalli Hills and Pollachi. These are the areas cultivating avocados. So avocados highly nutritious. It good for cardiovascular disease and it reduces the aging effect, and also it some cosmetic items it prepared from these particular avocado foods. So these avocados nowadays are exporting in Australia, from India to Australia. So 1 kg avocado fruit is 400 rupees to 500 rupees. So in this sense, the avocado production is inhibited by 4 different major pathogens. First one is Phytophthora cinnamomum, another one is anthracnose, stem rot, and Fusarium dieback disease. So in this particular presentation, we only focus on Fusarium dieback disease and shortwood borer infection. These are the major diseases infecting avocado. The first one is stem rot. It's a due to downward drying of the seedlings. Another one is a Fusarium dieback. This is the Fusarium dieback. It causes the disease in nursery and also in the field condition also. This is the symptom it exhibited by the Phytophthora species. This is the symptom also exhibited by the Phytophthora species, and also they are causing the disease in fruit also. So that these are the diseases it causing the yield reduction in avocados. So So in case of we are only focusing on Fusarium dieback and short-bowed borer. So in the disease occur in nursery condition and also field condition. In this nursery condition, the grafted seedlings it caused by the Fusarium disease. In this condition, the insect enter into the grafted region of seedlings. So here the avocado is promoted through two methods. One is the seed-based propagation and another one is the grafted seedlings. So in the grafted region, the insect is entered. So along with the Fusarium disease. So after these insects again enter into the field condition, it means trees. So there is a mated female insect. This mated female insect to making the tunnels, it means cavities and tunnels inside the these mature trees and making the small cavities. So in these cavities, the insect producing egg, larvae and pupae and adults. So again, they are male and female made together and finally the pregnant females come outside. So these insects again go and infect the new crop. So in this particular concern, we fix in the objective survey and assess the incidence of Fusarium dieback pathogen and software borer incidence. And also isolation of the Fusarium dieback pathogen from the infected seedlings and also the pathogen isolated from the insect. So finally, the morphological molecular confirmation of the pathogen and also insects. So and what is the mechanism behind the symbiotic nature also improved from these particular studies. During the surveys, their surveys are conducted in major avocado growing regions in Tamil Nadu. So in this condition, the study was in Kodaikanal, it reported major disease incident and disease severity and maximum failure of the grafted seedlings. It means we are grafted 100 seedlings, the 35 seedlings will be failure. It means one seedling costs 100 rupees. You think like how much the failure will be occurred. So in this condition, we have isolated the pathogen from infected in nursery condition, grafted seedlings and tree symptoms. So this culture again morphologically confirmed based on this is a correlate, a confirmed is a Fusarium. So these culture again to sequence using the IDS 1 and 4 and the sequences help of sequencing for biochart. So this sequence is a Fusarium evaluation. This is confirmed by this particular NCA database. Again, the pathogen is isolated from the particular insect. So this is the symbiotic nature. So this insect having the mitochondria, this is on the internal mandibular organ that transport the pathogen from this internal organ. So from the one tree to another trees. So their pathogen cultivated from this particular insect. These also convert as a Fusarium evaluation. So these pathogen again controlled by GCMS results. It's what are the metabolites inside the pathogen which will make the symbiotic infection of the insect and also seedlings. The pathogen also confirmed as same analysis is the producing macrocardia and mitochondria and clambrospore. So these are the pathogenicity tests conducted under the pot culture condition. So in this condition, the grafted. So this is the avocado seedlings grafted by science. Rootstock, the cyan is dipped in the pathogen culture and again grafted. So in this condition, all the seedlings will be expect the type of symptoms. So this the Fusarium will TKD 30% it expect the more disease incidence, disease severity 47.13%. And also the pathogen is fed by the insect. So this is a symbiotic nature. This insect feed this fungus. Again, this insect inoculated on the healthy seedlings. It make the some code on the seedlings and to make the diverse symptoms. It means the insect carry this pathogen in the interbody condition and to exhibit the diverse symptom and also the insect. So another one important the proved experiment for the insect enter into the mature trees. This is the insect, they are going inside the trees, they are producing some insects, like Fusarium producing some insects. Like the lactase insect, these insects making the cavity. So it made up of between this lactase to inhibit or digest the wood cavities to making the coal for to the insect multiplication. So in this the virulent isolate to exploit the more lactase activity 0.1521 units per ml. The pathogen culture to for GCMS analysis, which culture executed the three type of compounds. One is polyp. This compound is responsible for the symbiotic infection of Fusarium and Fusarium pathogen and also insect. And also pollen is food for the pathogen. It also importantly this pollen is also naturally present in the avocado and also present in the insect. So that's why the pathogen easily infect the avocado and also it carried by the insect. Another thing is the wilstic acid and palmitoleic acid and palmitic acid. These are the two-way interaction. One is the insect and the pathogen to what the mechanism of symbiotic transport of these pathogens and insect. This is the lignoceric acid, veganic acid, arachic acid. These are the three compounds, it responds to a three-way interaction. It means host, pathogen and insect. So in this condition, the host responsible for the attraction of the insect. So this is the pathogen Genomics analysis, and also the Genomics analysis of Fusarium infected cell that excreted the beta transparacin. This compound, it naturally attracts the insect to easily invest on the host. So finally, I conclude that is no more doubt about this particular study. So the pathogen and insect is symbiotically associated, and these compounds are excreted by this Genomics analysis. It proved by this symbiotic oriented compounds. So these the future prospect for these particular studies, we will to isolate some endopeptic agents that will repel this particular volatiles and metabolites. It will reduce the attraction of insect, and also it colonize inside the insect to reduce the incidence. Thank you.
It's open for discussion now.
You mentioned about the study and squalene. How we can use that research findings to manage this both?
In these conditions, the Genomics analysis of infected seedlings executed the trans-beta-panacin. These compounds attract the insect. So we are using any biocontrol agent to reduce the particular reduce or repel that particular compounds. So automatically we reduce the insect attract. Another one thing, we are using the biocontrol agent which colonize inside the insect, like as the evaluation communicators. So automatically reducing.
How you have established that this beetle is in association with this fungus?
We have proved through pathogenicity testing. The pathogen is isolated from the infected insect.
You have dissected the insect?
They are having the one particular organ, it's mitochondria. This is the internal mandibular organ, that organ only they carry the fungal parasites. So we are only focusing on that particular region.
What region?
Mitochondria. Is present in only the evolution groups. Evolution which part of it? It's upper part of the particular mandible. It's great.
Who is the entomologist associated with this study?
Jairus. Mother Jairus.
Because most of the short-haul bowlers,
they are also
dried twigs or dried part of the twigs.
But we have collected the dried, is not that no any normal eggs people not is there. Is not present. Only the somewhat healthy plants only having that insect.
Is this the first?
The first.
Actually, avocado is
It is very difficult to identify this problem. So I was at very well campus and also not able to test this carcinosis, whether it is a pest or disease, it is a controversy. So your work simply explained that it is a Fusarium with gold. So what is the mechanism of Fusarium?
Here Fusarium explained the three more mechanisms. Lactase enzyme, it produced by the Fusarium, that making the digest of the wood cells inside the menstrual trees, it making the cavity, that cavity useful to the insects. Egg, larvae, pupae. So they complete the life cycle within the 30 days. Again they come outside into the new growth.
Actually, generally we are having a mechanism of vessel plating and
apart from this mechanism executed by this Fusarium evolution.
It is verified this literature.
Yes, sir, verified, sir. Some literature also reported the lactase enzyme. This responsible.
So you can write it for natural or renewable.
Yes, sir.
Only say that it is dieback symptom. Only dieback symptom is associated with workout. You say it always after sleeping.
Yes, sir.
Only say that it is
in this condition, we are using the cyan material for propagation, sir. So in this condition,
first time I am hearing.
Fusarium factors. Many literature in California, many articles about Fusarium bicarbonate. So the graph there, the cyan material transmitted by the Fusarium. Any Fusarium imported series, they are using the signature for rapid purpose. They are easily transmitted.
Then what is this?
It's a second topic, sir. I also isolated the semi-truss and the pelvic from this same seedling, but they are not in travel seedlings, only in seed-grown seedling.
There is also associated.
Associated, sometimes associated.
Maybe the reason.
Yes.
So have you observed this sun blotch viral disease in our country?
Yes, in that we say region, no sun blotch.
It is not there.
Yes, we are using the PMPs and the use the protocol any
In where it is available? That is not in
literature. I will go to literature, but there is no reporting in India. India.
So very nice, very good observation.
Thank you.
Thank you, Mr. Ayandure, for your valuable information. Now I call upon R. Sariga to present on the topic genetic acid.
Titled on genetic acid associated with hemoglobin disease in acid plant. So acid lamb is one of the important commercially cultivated crop in India. In this, the most devastating disease is citrus greening disease. It's otherwise called as hemoglobin disease. It is often confused with symptoms. It is often confused with symptoms of nutrient deficiency. So farmers recognize later it was infected with pathogen. Then only they are approached for the pathologist. So detection is more challenging due to low concentration and also uneven spatial and temporal distribution of the pathogen in plants. So in this plant pathogen system, when citrus greening is infected plant, the accumulation of 2,5-dihydroxybenzoic acid, that is gentisic acid is accumulated, which is restricted in the systemic non-necrotizing infection in acid line. So this 2,5-dihydroxybenzoic acid is act as an intermediary signal molecule in systemic acquired resistance. So as we all know that aromatic benzene ring compounds are very much essential during the plant biotic and abiotic stress, and they constitute a major part in plant's defense mechanism and for also the production of secondary metabolite and play various role in physiological and mechanical activities. In this picture, the salicylic acid biosynthesized is produced in pathway of isocorismate synthase pathway, and also by means of phenylalanine ammonia lyase pathway. As we all know that after salicylic acid production, downregulation of salicylic acid will take place. After that, hydroxylation and oxidation of salicylic acid will take place. It will produce the gentisic acid, which means 2,5-dihydroxybenzoic acid. It acts as an antimicrobial compound and also the signaling system for activation of inducible defensive plant systems. This picture, I would like to explain that in healthy plant also gentisic acid is present. How we differentiate means the presence of or amount of or concentration of gentisic acid is used as a marker for identification of the citrus greening disease. First of all, in infected plant with response to pathogen, salicylic acid production will induce. That salicylic acid will produces the gentisic acid. That local concentration of the gentisic acid will induces the upregulation of the defense regulator genes like NPR1, PR proteins like PR1, PR5 or PR2. This will induces the systemic acquired resistance, which is called as a long-term immune memory, and it will produce the host plant resistance mechanism. I will take healthy leaf samples and also infected leaf samples. Then I analyze the qualitative and quantitative estimation of gentisic acid by qualitative estimation by means of TLC and quantitative estimation by means of spectrophotometer. The standards were purchased from Sigma Chemicals. The internal standard which I was used was 2,5-dihydroxybenzoic acid, 99%. This is analog of the gentisic acid. Concentration of 0.05% to 1 milligram per ml. From this, the result collected by gentisic acid was detected as a marker element because while comparing with the control, the band size of CLA's infected samples were similar to that of that standard, and the healthy leaf samples contain the lowest concentration of gentisic acid. In same The same results were obtained from the quantitative estimation also. Here the quantitative estimation is carried out by means of a UV spectrophotometer with the gentisic acid standard. In this, the infected samples contain higher amount of gentisic acid. From this I conclude that gentisic acid is a pathogen inducible signal. And also, it was also called as a hydroxylated salicylic acid, which was characterized as an antimicrobial compound and signaling system for activation of inducible defenses. Although this gentisic acid was assumed to be because it was present in both healthy and also in resistant cleaning infected plants, but its specificity will future it will be characterized and it will be used for the used to control this pathogen in future days. Thank you.
Is open for discussion.
So, which is the hotspot for greening, citrus greening in India? Is in Tamil Nadu?
So, most of the south belt of Tamil Nadu was heavily affected with the citrus greening disease, sir. Especially in Tenkasi district, the moreover plants were declining, sir.
Okay. So, how your research finding will help the farmers?
So, gentisic acid was available as a chemical in a Sigma chemical, sir. It was cost was about 2,000 rupees, sir. But farmers are not preferable to spray the chemicals. They are giving a nutrients sprays and because it was similar to zinc deficiency, sir. And also the nutrient transportation also stopped because of this cure long wing disease, sir. So we prefer more nutrient application for plant with support to our bacillus and normal our biocontrol agents, sir.
During your research program, did you explore how this disease is managed in other countries, especially citrus growing countries?
Sir, in Florida, they are also injecting antimicrobial peptides in trees and they are screening, sir. But effectively controlling, they are trying to control the vectors, silly. Because we can't able to manage this disease in trees, sir. We can manage by means of vector control only we can manage.
How will you diagnose this disease?
So we diagnose by 3 different needs, sir. Biochemical, primary metabolites, secondary metabolites, and through molecular characterization, sir. By primary metabolite was characterized.
Molecular characterization, what is the method?
Sir, normal DNA extraction and following specific primers. Using specific primers, we characterize.
What about lamp?
So lamp also we characterized for preliminary screening. So for pathogenicity studies, we use lamp studies.
When the lamp is applicable for this disease diagnosis. So, sir, I'll already asked one important question. How do you manage this disease?
Sir, we can't manage by means of applying any other chemicals, sir. We are giving nutrient supply more to plant to balance the disease, sir.
Actually, foreign countries they are managing this disease. Just to look up the research findings and also work for that line because this acid and something it is costly and it is not applicable to the farmers.
Thank you, Ms. Sariga, for your mind-blowing presentation. Next, we call upon the presenter, Ms. M. Jayaveni, to present the topic, Influence of Microbial Inoculants on Resistance in Bendy Accessions against Shoot and Fruit Borer, Areas.
So as we all know, Bendy is nutrient-rich crop grown in tropical and subtropical regions of the world. It is mainly tapped by many insect pests, which commonly include shoot and fruit borer, that is Aedes vitella, that encounters both in vegetative and reproductive stage. So these are the various biostages of Aedes vitella. So why there arises a question where why there is a need for induced resistance, and as we all know, there are alternate approaches as being arising as a paramount to avoid the dependence of pesticides. One such invention includes the identification of wild and cultivated donors for shoot and fruit borer, that is Salem local and Artha anamika, which in turn they shows resistance and those developmental resistant varieties are found to be ideal component of IPM. Major objectives include to evaluate the induction of resistance and to study the various mechanisms of resistance and also to study their biophysical and biochemical factors.
So these are the various microbial inoculants being used for the study, which includes azospirillum, phosphobacteria, Pseudomonas, K. solubilizer, brown algae, green algae, and red algae, with their respective dosage and days of applications being carried out. Initially, field evaluation is being carried out to analyze the fruit infestation, in which the bendy ascisions, both Salem local and Arta anamica, is being sprayed with various microbial inoculants, and then the infestation is being studied. In the first mechanism, that is anti-zoonosis, here it is being it is been identified by using by analyzing feeding preference and the oviposition preference. In case of feeding preference, where both the ascisions, the fruits are being collected from various microbinoculant spray and placed in a tray where randomly the larva is being released. In case of no choice test, tunneling damage is being measured. Ovipositional preference, 45 days old treated plants, microbial inoculants treated plants is being placed in cage and adults has been released to check their egg laying performance. In case of antibiosis mechanisms, the length of the larvae, weight of the larvae, length of the pupa, larval period, pupal period has also been studied. In case of biophysical base of resistance, in which fruit bits is being cut from various bendy ascisions sprayed with microbinoculants, in which these fruit bits is being boiled for 20 minutes and then ethanol treated for the growth, for the removal of chlorophyll content, and then microscope studies has been carried out to know about the trichome density, length and breadth in respect to pest damage. Biochemical bases include the total phenol estimation, sugar, nitrogen, phosphorus, potassium, ash content. In which the research shows that K. solubilizer, which as we all know, the potassium and phosphorus found to be in part resistance to pest when applied, and hence the research also verified that K. solubilizers were found to be shows least fruit infestation when compared to brown algae, which shows the highest fruit infestation. In case of a biochemical basis of resistance, as we all know, sugars tend to attract the pest, in the insect pest, and hence the K. solubilizer found to be possess at least reducing sugars and phenol content. In case of the economic character also, K. solubilizer also increases their number of fruits per plant, single fruit weight and also fruit yield per plant when compared to brown algae, where there is reduced yield. And when comparing the correlation between the trichome density, length, breadth was found to be negatively correlated with the fruit damage. In case of biochemical characters, when compared with the fruit damage or oviposition preference, sugars and nitrogen are found to be positively correlated, whereas phenol, phosphorus, potassium was found to be negatively correlated. Thank you.
Again, same common question, how you will translate to the farmers' benefit?
So rather than being applying various chemicals to rectify the first problem, we can apply the microbinoculants as to show rectify the system.
Okay, so microbial inoculation means, did you identify anything for that or?
No, sir. Commercially available microbinoculars are being used for study.
Okay. So, what is the K. solubilizer? You mentioned that is the beneficial one, right? So, can we exploit that commercially?
Yes, sir. It's a thank you artificial has been commercially available. It's also available.
No, I'm talking not from the. I'm talking from the chemical perspective because I'm talking chemical perspective. So, from that angle, is there any possibility? So, because sometimes the direct concentrations actives will be helpful. So from that angle, I thought of understanding about this particular one. Okay, thank you. It's slides are nicely arranged. So how did you select the treatment?
So the most commonly available.
Is there any report about this microorganisms, these biofluxes, what is the mechanism?
The mechanism is involved. So as I said before, the potassium and phosphorus
also found to be impact resistant to.
What about others? You are choosing so many microorganisms, bioplaces. What is the
So
role?
some of them are root inhabiting microbial organisms. Where they also increases the plant growth also.
No, we are, this is against the pest. So how it is related to managing pest? What is the role of biofertilizer in managing this pest? Okay, thank you.
So as raised by the chairman and the co-chairman, definitely it can be an incidental observation. So we are applying microbes or biofertilizers and we are directly relating them for the growth benefits of the crop. And incidentally, they may impart resistance. So they are not directly involved in pest management. It is an incidental observation. That is my conclusion. So if you fully understood the rationale behind the selection of your topic, you will be able to explain.
We invite Ms. M. Chanani to present the topic Management of Root Knot Nematode Meladogene Incognita using Pasteurial Penetrance in Tubercle.
This is the outline of my presentation. It deals with the contents about the crop tuberose and what are the major threats causing in tuberose production and how the root knot nematode Meladogene incognita is causing infection to the crop. A control measure has been taken against this was using a biocontrol agent that is Pasteurial penetrance. By using this, a water-soluble formulation was developed and tested in microplot conditions. So first, coming to the crop tuberose, it is one of the most important, significant tropical ornamental bulbous flowering plants. They are known for the long-lasting flower spikes. And another important aspect, it has 4 different types of flowering. One is single, semi-double, double, and variegated. In this study, the hybrid used was Arca Ridgewall, which belongs to single type. The area under tuberose cultivation in India is about for 95 lakh hectares. Though the area under tuberose cultivation is commenced in many regions of Tamil Nadu, the potential productivity has not been achieved so far. So this was mainly due to the influence of many pest and diseases. Among them, the root knot nematode Meladogene incognita is the most destructive pest damage causing to the crop. So this nematode feeds as a sedentary feeding behavior and it undergoes 4 zonal stages, that is from J1 to J4. Here J2 will be the infective stage and the J2 gets migrated to the cortical tissue. So there they will establish a permanent feeding site called genicle and this is the ecmasis which is released from the infected root portion and the male nematodes they become vermiform after feeding and the females start to reproduce. So due to this drastic damage caused to the crop, it results in the stunted appearance and loss of flower production. Coming to the symptoms, the above ground symptoms like stunted patches in plants and loss of flower production, and below ground symptoms like decreased size of tuber formation and moderate to severe galling in roots. The gall and ecmasis are formed in infected root area, and after complete feeding, the females attain a globose-shaped body. Why Pasteurial penetrance has been used as a control? Biological control agents of plant parasitic nematodes are typically very specific natural enemies of the target plant pest nematode. So one of the best studied biocontrol agent is Pasteurial penetrance. It is an obligate parasite of root knot nematode Meladogene incognita. Its mode of action, it will inhibit the nematode from producing eggs. The endospores of Pasteur will get attached to the cuticle portion of the nematode body. So the life cycle, when the nematode penetrates into the host root and feeds, there they will develops a germination tube. So this tube will pierces the cuticle of the nematode and forms mycelial-like popcorn shaped balls. This mycelial popcorn balls are of several structures that are formed in Pasteur during sporulation. So the ovarian tissue of the nematode will be filled with endospores which will replace the nematode eggs. For example, a healthy root knot nematode can lay up to 1000 eggs in the soil, whereas a Pasteur infected root knot nematode does not lay any viable eggs. So this is the mature endospore. And this is how the endospores of Pasteur get attached to the cuticle portion of the nematode body. By using this bacterium, we have attempted with an eco-friendly water-soluble formulation. Initially, the sterile water was heated, and upon that, emulsifier, surfactant, glycerol, 280, 220 were also added to this mixture and was heated for about 3 minutes. Finally, the endospore suspension of the Pasteur were added. This is the developed water-soluble formulation. And this is the infested females which contains the endospores, and this was the stability of the endospores in the developed formulation. So based on the results of pot culture experiment, which was conducted as a basic study, the optimized dosage has been selected and practiced in microplot. Here two treatments were maintained with each 13 replications. Healthy tubers of tuberose were planted in all the replications, and healthy plots were made into sick plots by inoculating the root knot nematode zoonoids. Then treatment plots were given a concentration of developed water soluble formulation 2 ml per plant, and the control was left as such. After 45 days of interval, the observation has been recorded, and the results clearly indicated that the number of flower production and the overall plant density were more in treated compared with control. So the root also gained more in treated compared with control. There were severe gall infestation and stunted appearance. So the plant biometric characters such as shoot length, shoot weight, number of stalks emerged, flowers emerged were comparatively more in treated than control, and the flowers produced in control were less small in size, which lost their marketable value. The gall and igmosis formation in treated plot were reduced compared to control, and the galls in treated does not contain any viable eggs. We also collected the infested females in the formulation treated plant. So the root biometric characters also came up with the same trend, with showing a more number in treated compared with control. Here in untreated, there were decreased size of tuber formation compared with the treated plot. Hence, the plant and root biometric characters shows more potential in the formulation treated plant. And concluding with, since there are many existing successful biocontrol agents against plant parasitic nematodes are available as commercial, Pasteur is a very potential bacterium against root knot nematode Myldogene incognita due to its obligate nature and low specificity. So, in accordance to this, an alternative approach has been taken by using this bacterium and is tested in various aspects. So, it has reduced the ecological impact of using chemical treatments.
What's the duration of this tuberose crop?
Sir, averagely it will take 4 to 7 years. They will grow.
Okay. So you conducted in a watered experiment.
Yes, sir.
So now you already mentioned it's a long duration,
Yes, sir.
multi-year crop.
Yes, sir.
How you will recommend this, and what stage it should be recommended?
Yes, sir. Sir, here a basic study pot culture was conducted, and based on the results of that only we have carried out with microplot. Field trial also were conducted with the same formulation. Due to short span of time, only one season data we have observed. In that, they have documented the presence of confirmation of the Pasteur endospores present in the females of nematode body.
My question is here. Okay, so you proved that this
Yes, sir.
efficacy wise, I'm not.
So one more season may be required to taken into a broad spectrum, sir.
No, not the spectrum also. Okay, it's controlling very well, 100% efficacy, etc. So here you have done in the potted experiment.
Yes.
What's the timing of publication? When you translate this study to the farmer, which is going to happen in at least 5 to 6 years time frame. So what is the critical window you will be recommending? Because these nematode once enter the roots.
Yes, sir.
So you can't control that one.
So no, sir. Pasturia it is a potential bacterium. So maximum it can take at least 1 year for controlling all the generations of the nematodes, sir.
Okay, so that means you will be recommending multiple timing of application. If yes, what's the interval?
So that only, sir. The lagging thing is mass production is lagging. So if we concentrate on that and any alternate mass production methods are
no, mass production also I will give you.
Okay, sir.
So what's the recommendation window? Okay, say for example,
yes, sir.
rice stem borer, there are 2 different windows. One is the vegetative phase stem borer, degards, and then white ear. So at least minimum 2 application we need to do in a hot spots where it is both the generations are there. Similarly here, it's a multiple year.
Yes, sir.
What's the critical window you will be recommending so that the farmer will get benefit of your product? Don't worry about the mass production, I will give you.
Yes.
Whatever the quantity, if you want, I will give you. So how will you recommend to the farmer?
So my answer will be, I have conducted the field trial. So based on that I have experienced, if we are seeing for 2 more seasons, I can get an idea of what is the recommendation, sir.
What exactly you
now
are
I can't.
looking? Okay, you decided that you want to do for 2 more years. So what you will be looking for in the next 2 years?
So that I can practice and things.
Okay. So I will give you an answer.
Yes, sir.
So you need to protect the new roots.
Yes, sir.
Try to understand the rooting pattern of the crop. How exactly it's happened. Is based on irrigation or based on the rainfall or based on season. So you may need to program your application in that.
Yes, sir.
Thank you.
Yes, thank you.
So already you have conducted part culture and microcrop studies.
Yes, ma'am.
So in those studies, how you have done the applications? So that is the answer for this question. At what time you have done?
Time.
Time was the crop.
When you have applied?
10 days after planting or 10 days.
So is it a blank
28 days,
application?
28 days,
I
I have sown at only tubers, sir. After it had taken 28 days of planting, I have inoculated my formulation.
So it is symptom based.
Yes, ma'am.
You have observed the symptoms
Yes,
and simultaneously you have taken the samples, you observe the population,
Yes,
then you have done the application.
In microplot condition, we have made the leafy plots into 6 plots and then the formulation was treated. Because it was in an empty state, so we have to in a condition to infest the plot, then we carry out with the formulation.
Or is it possible to apply based on the symptoms? So do you have different grades by which you can identify the level of infestation?
Symptoms, above ground symptoms, we can't detect this root knot nematode. Only foliar nematode like Aflancodus busy is also a threat causing nematode in tuberosome. So based on this nematode, only we can see the stunted patches and the overall flower productivity. Then we can come to a conclusion that this infestation is by this nematode.
Then you can go for spot application also.
Yes, ma'am.
Okay, thank you.
Thank you.
Thank you, Ms. Janani, for your delightful presentation. We call upon the next presenter, Ms. M. Danvarsha, to present the topic SNP analysis and in silico docking studies on serotonin pathway for yellow stem borer resistance. She is not available here. So we move on to the last presentation of this theme by Ms. C. Muthulakshmi Babitra on the topic transcriptional factors regulating insect development and insecticide resistance.
Good evening to one and all present here. Today I am here to present about the topic transcription factors regulating insect development and insecticide resistance. We all know about the central dogma of life, which involves mainly of two processes. One is transcription and another one is translation. Transcription is a process where the DNA being to an adult. I am here to present about the topic transcription factors regulating insect development and insecticide resistance. Coming to the introduction, as we all know about the central dogma of life, which involves mainly of two processes. One is transcription, where the DNA is copied into RNA, and the next one is the translation, where the RNA produces the protein. So, in the transcription factor is very much essential. Transcription factors are nothing but the protein which regulate the transcription of the genes by binding to the enhancer or promoter regions of the DNA. Transcription factors contains the DNA binding domain, which will specifically recognize the transcription binding site in the DNA, and we regulate the transcription by regulating the RNA polymerase enzyme. So these transcription factors make up approximately of 3.5 to 7% of the coding sequences, and they are involved in various diverse biological functions. They are generally classified based on the DNA binding domains. These are the major structures present in the insects. They are of three superclasses. One is the basic DNA domain, and second one is the zinc finger DNA binding domain, and major one is the helix-10 helix, which contains of different homeodomain structures like Hox protein, PAG-like protein, PAG domain protein, POE protein, and LIM protein. So these transcription factors are involved in various biological functions, and they respond to both internal responses and external responses. Under internal responses, various functions and their transcription factors regulating them are represented in the table. For example, in the embryonic axis development, the transcription factors that is bipart, BCD, and hunchback that is HP, they regulate the formation of the antigen region, and the transcription factors nanos and caudal region, they form the posterior region of the embryonic axis. In case of external responses, they are divided into biotic factor responses and abiotic factor responses. In case of biotic factor responses, they will regulate the immune responses genes, like when the pathogens or any